Good afternoon, everyone. I'd like to begin by thanking the organisers for giving me the opportunity to speak at this fantastic conference. My name is Andrew Lawson, and I'm a postdoctoral fellow in Inigo Martin-Craner's lab at the Wellcome Sanger Institute, where I previously carried out my PhD. Today, I'm going to talk about the work carried out in our group over the past decade, investigating the role of somatic mutations across health and disease. As many of you will be aware, the cells in our bodies are not genetically identical to each other. From the moment the zygote first divides, each cell in a developing embryo, and subsequently our bodies, continually acquires mutations that are private to that cell and its descendants. These somatic mutations arise due to the intrinsic imperfect fidelity of our DNA replication and repair machinery, although the rate at which these mutations are acquired can be accelerated by exposure to mutagens in our environment, such as ultraviolet light and tobacco smoke. Now, most of these mutations are tolerated remarkably well by the affected cell, with relatively little impact on its phenotype. However, occasionally a mutation occurs that allows the cell to divide faster or survive longer than it otherwise would. These mutations are known as drive mutations, and when a cell acquires on the order of 5 to 10 such mutations, its behavior becomes sufficiently aberrant that it has been transformed into a cancer cell. Back in 2000, the Cancer Genome Project was established at the Wellcome Sanger Institute by Mike Stratton, and its primary goal was to characterize the highly rearranged and mutated genomes of cancers that are the culmination of this decades-long evolutionary journey. However, more recently, my primary research focus, along with several other groups at Sanger, have shifted more towards earlier steps in this process, specifically how cells in histologically normal tissues outcompete their neighbors when they acquire their first drive mutation. Although cancer genomes are often grossly rearranged and they can deviate substantially from the human reference genome, detecting somatic mutations in normal tissues is a more difficult technical challenge than detecting them in cancer biopsies. The reason for this is that in cancer biopsies, we're able to distinguish cancer cells from neighboring normal cells using histological markers. This means we can isolate cells that share the same clonal mutations, and we can then use the consensus of the DNA sequence from many cells to confidently call somatic mutations. By contrast, in normal tissues, we often cannot easily distinguish clonal units that share mutations, and even if we can, the size of these clonal units are considerably smaller than those in cancers. This means that if we carry out bulk sequencing of a normal tissue sample, the vast majority of somatic mutations will affect a proportion of cells less than the detection limit imposed by the standard sequencing error rate, which is on the order of 0.1 to 1%. Therefore, we would only detect a handful of somatic mutations that reach high enough variant allele fractions due to having occurred very early in embryogenesis. Therefore, in order to identify the full complement of somatic mutations in a normal cell, we need to use specialized strategies to overcome this issue, several of which I'll talk about in this talk. The first approach that we use to allow us to identify somatic mutations in normal tissues is deep targeted sequencing of small microbiopsies with an area of approximately 1 millimeter squared. The first tissue we analyzed in this way was peeled epidermis from eyelid samples obtained via blepharoplasty. This strategy relies on mutant clones reaching a large enough size that a proportion of mutant cells that carry mutation exceeds the detection threshold of standard Illumina sequencing, as I said before, having a variant allele fraction of greater than 0.1 to 1%. We found the results of this initial study to be truly remarkable, and in many ways this has paved the way for much of our subsequent work. So this square represents a 1 centimeter by 1 centimeter piece of human skin, and each circle indicates a clonal expansion, with the color denoting the presence of a drive mutation in one of the indicated genes. This revealed that despite appearing entirely histologically normal, our skin is actually a mosaic of microscopic mutant clones. We estimate that by late middle age, epidermal cells in sun-exposed skin have acquired on the order of 20,000 mutations, mostly caused by ultraviolet light radiation, and that around 25% of cells have acquired a drive mutation. The identity of these drive mutations closely matches those observed in squamous cell carcinoma. In particular, drive mutations in the Notch signaling pathway are believed to alter the balance between stem cell self-renewal and differentiation in epithelia, providing an advantage over long-term to those mutant cells. A question that arose from the study was whether these mutant clones are an unusual property of skin due to the high somatic mutation rates caused by exposure to UV light, or whether all tissues are a similar patchwork of mutant clones. We therefore followed up this initial study by carrying out an analogous study in normal human esophagus. We used a similar approach of peeling the epithelial layer from the underlying submucosa and cutting it into small microbiopsies. This cohort included a substantially larger age range of individuals, and we observed that mutations accumulate linearly with age. Although this is now well established across many tissues, it was an important result at the time, as some models of cancer evolution hypothesized that there may be supralinear increases in mutation rate with age. The mutation burden in epithelial cells in human esophagus is approximately an order of magnitude lower than sun-exposed skin, i.e. around 2,000 mutations per cell. However, despite this lower mutation rate, we actually see increased evidence of selection in esophageal epithelia compared to skin, with around 50% of cells possessing a drive mutation by middle age. As this plot demonstrates, we clearly see that the driver density increases with age, but it can also be affected by other factors. For example, the 45-year-old male who's a heavy smoker on the left of the middle row has a driver landscape similar to that of non-smokers in their 70s on the middle and right of the bottom row. One particularly striking result from the study was that the proportion of cells that possess a drive mutation in Notch1 is in fact substantially higher in normal esophagus than it is in esophageal squamous cell carcinoma. This is in stark contrast to the behavior of P53, which shows a much higher degree of enrichment. Sorry. Okay, sorry.
So, I think we should just reload this. Yeah. And then come back here. Just move one slide on it. Okay.
Okay. So, rewinding slightly, one particularly striking result from the study was that the proportion of cells that possess a drive mutation in Notch1 is in fact substantially higher in normal esophagus than in esophageal squamous cell carcinoma. This is in stark contrast to the behavior of P53, which shows a much higher degree of enrichment in esophageal squamous cell carcinomas than normal tissues, as we might expect for a gene commonly referred to as a gatekeeper of cancer. This strongly suggests that inactivating Notch1 provides a strong selective advantage in the context of normal esophagus, but that these Notch1 mutant clones are actually less likely to progress to cancer than wild-type clones. This result has been followed up using mouse models by Emily Abbey, a PhD student in Phil Jones's group, and her results are concordant with this hypothesis and were published in Nature Genetics in 2023. We further improved our strategy for studying normal tissues in our next project, investigating bladder urothelium, which was the main project I carried out during my PhD. We instead used laser capture microdissection to isolate short stretches of urothelium, the lining of the bladder, and developed a modified library preparation approach to enable sequencing of these low input samples. I microdissected on the order of 3,000 such pieces of urothelium, of which 2,000 yielded sufficiently complex libraries to perform targeted sequencing. And we also selected 88 sufficiently clonal samples for whole genome sequencing. As with the esophagus, we saw a linear accumulation of mutations of age. Of note, the mutation burden was approximately half that of esophagus, and the proportion of cells with drive mutation was around a quarter of that of esophagus, around half that of skin. Using this microdissection approach, we were able to map the proportion of cells in each strip of urothelium that possesses a drive mutation. In this sample from a 59-year-old male, you can see that we detect 6 different ARID1A mutations in this around 1 centimeter stretch of urothelium. By contrast, this sample from a 53-year-old female has 7 different KD6A mutations and no ARID1A mutations along a similar length of urothelium. When we formally analysed this phenomenon, we observed a remarkable preference for certain drivers in some individuals, with those squares highlighted by blue boxes indicating significantly stronger selection in that donor compared to all other donors in the cohort. It is also worth noting that the drive landscape in bladder urothelium is notably different to skin and esophagus, with a preference for drive mutations in epigenetic modifiers, such as the histone methyltransferase KMT2D and the histone demethylase KDM6A. No worries.
So sorry. Maybe we have to close. Where is it? This. This. Okay. This is it. So just quickly want to. Is it moving? Yeah, no.
Go ahead. Okay. I think.
Sorry about this.
Is that? Sorry. Where are we up to? Okay. So, as I said, the driver landscape in bladder is somewhat different to other tissues with a preference for epigenetic modifiers. This first part of the talk has raised several important questions. Firstly, all three of these tissues were epithelia, which we studied as the majority, around 85% of adult cancers, are epithelial-derived carcinomas. Do we see a similar patchwork of mutant clones in other tissues? Secondly, what is responsible for this driver preference across individuals? Is it genetic background, mutagenic exposures, other lifestyle choices? Thirdly, do individuals with a particular driver preference have a higher or lower risk of developing cancer? Fourthly, is it possible to manipulate the driver landscape of normal tissues to disfavor progression to cancer? Can we develop something equivalent to statins for cancer? For example, would we reduce rates of esophageal squamous cell carcinoma if we increase the selective advantage of NOTCH1 mutant clones and reduce the selective advantage of p53 mutant clones? Finally, the role of somatic mutations is well established in cancer, but do these somatic mutations also have a role in deterioration of tissue function with age and in non-cancer diseases? In order to address these questions, we needed to turn to other technologies, as microdissection is both low throughput and limited for use, and limited samples with spatially related clonal expansions. One alternative approach that has been used by other groups is to generate single-cell derived clonal organoids. This approach has several advantages. You can guarantee that each sample you sequence will be clonal. You can select particular cells of interest using cell surface markers, and there is no need for neighboring cells in the original tissue to be clonally related. However, there are also several disadvantages. The culturing conditions may need to be optimized per cell type and only works for cells that can be expanded in vitro. Any mutations that are required in vitro prior to the single-cell bottleneck will be indistinguishable from genuine mutations, and the process is still relatively low throughput for assessing the driver landscape of an individual, often needing hundreds of colonies per donor. Nevertheless, this has proved a particularly powerful tool for understanding the accumulation of mutations in certain cell types, particularly in hematopoietic stem cells. In some beautiful work carried out by Emily Mitchell and Peter Campbell's group, the phylogeny on the left shows that many of the hematopoietic stem cells in a 29-year-old individual are unrelated, whereas the phylogeny from the 81-year-old donor on the right shows clear evidence of several hematopoietic stem cell clones taking over the bone marrow, a process known as clonal hematopoiesis. Intriguingly, canonical drive mutations were only identified in a small number of the expanded clades. Due to the limitations associated with clonal expansion of single cells discussed in the previous slide, we have instead focused on a different solution to the problem of how to scale up somatic mutation detection, which is error-corrected sequencing. Unlike the previous approaches, if we can reduce error rate of sequencing to be substantially below the somatic mutation rate, we can assess the acquisition of somatic mutations in many clones simultaneously. One of the most successful approaches for reducing the error rate of sequencing is duplex sequencing. This approach has been pioneered by several other groups, including the work of Lawrence Loeb, Michael Schmidt, Scott Kennedy, Rosanna Risquez, Margaret Huang, and others. The principle of this approach is that if you tag each individual DNA molecule with some form of unique molecular identifier, either a random barcode in an adapter, or you can use the start and end position of the fragment at low sequencing depths, then, due to the asymmetry of the Y-shaped adapters, we can infer whether a sequencing read is derived from the top or bottom strand of a given DNA molecule. In order to call a mutation, we require consensus between the reads derived from both the top and bottom strand. This, in theory, should reduce the sequencing error rate to be close to the square of the polymerase error rate. High-fidelity polymerases have an error rate on the order of 10 to the minus 4, 10 to the minus 5, so the error rate should be on the order of 10 to the minus 8 to 10 to the minus 10, which is below the somatic mutation burden of most normal cells on the order of 10 to the minus 7 per base pair. Well, that's the theory. However, when we assessed several of the existing duplex sequencing solutions, we found that they had a substantially higher error rate than predicted, often closer to 10 to the minus 6. The reason for this is that standard end repair and A-tailing in library preparation includes polymerases capable of copying information from one strand to the other, violating the fundamental principle of duplex sequencing. Therefore, we resolved this issue by including dideoxy BTPs, i.e. each base other than A, during A-tailing to prevent nick extension, and we also used approaches that produces blunt ends, so there was no need for 5' filling by end repair. Using this approach, we were able to assess the accumulation of mutations in post-mitotic tissues, such as neurons and smooth muscle. From this, we found that DNA damage is converted into double-strand mutations at a high rate in the absence of DNA replication. Using this approach, you can see that these post-mitotic tissues have a comparable mutation burden to granulocytes, which is a proxy of the accumulation of mutations within hematopoietic stem cells, and an order of magnitude higher than in gametes. If you are interested about mutation accumulation in sperm specifically, I recommend a preprint on medRxiv by Matthew Neville, a talented PhD student from Heli Rabari's group that has recently been accepted in principle for publication in Nature. Our initial nanoseq approach used restriction enzymes to fragment DNA, which can help ensure no information is copied between strands. However, this came at the cost of only achieving partial coverage of the genome, around a third of it. This is sufficient for accurate estimation of mutation burden and signatures, but it cannot be used for driver discovery. So we've recently developed an updated version of the nanoseq protocol with a comparably low error rate to the original protocol that has full genome coverage. One way in which we commonly assess the error rate of duplex sequencing protocols is to use cord blood samples. We know from both the original nanoseq protocol and single-cell-derived colonies that neonatal blood cells possess on the order of 60 to 80 mutations, and so they have a mutation burden on the order of 10 to the minus 8 per base pair. The plot on the left shows that other duplex sequencing approaches have error rates that substantially exceed this burden, whereas our new full genome approach estimates the burden correctly. Additionally, other duplex sequencing approaches have a mutational spectra with many C to A mutations caused by single-stranded oxidative damage in vitro, whereas our new approach obtains a mutational spectrum consistent with that observed in single-cell colonies. We also apply this approach to formalin-fixed samples, which have high levels of DNA damage. As expected, in the input sample, if the input sample has a high level of damage, standard duplex approaches that allow information transfer between strands end up estimating very high mutation burdens, whereas our new approach estimated a burden consistent with a formalin-free control sample. Hopefully, having demonstrated that our new nanoseq approach with full genome coverage gives consistent mutation burden and spectra, we now move on to its utility for identifying driver landscapes. This plot shows the driver genes identified in normal bladder during the laser microdissection project I carried out during my PhD. And this second plot shows what we obtained by incorporating a targeted bait capture step in our full genome nanoseq protocol. And we faithfully recapitulate the previously observed driver landscape in normal bladder. We already knew this landscape, so why is this exciting? The reason it's exciting is that the plot on the left required 3 years of painstaking work to generate, whereas the plot on the right required just 1 week of relatively simple DNA extraction and library preparation. This means that when we sequence a single polyclonal population of cells, we can simultaneously detect hundreds of clones. The probability of a clone being sampled is proportional to the size of that clone, and this means that we can now study driver landscapes at an unprecedented scale. For the rest of this talk, I'll be discussing the initial project we've carried out to demonstrate the utility of this high throughput approach for interrogating driver landscapes. We asked over 1,000 volunteers in the Twins UK cohort to provide buccal swab samples for analysis by targeted nanoseq. Additionally, we also analyzed 371 archival blood samples from a subset of the donors. This flowchart shows sample attrition at various stages in the project. Entertainingly, in 132 cases, we could likely identify what the volunteer had likely eaten for their previous meal, as we had to exclude samples due to high levels of either chicken, pig, or cow DNA contamination in the buccal swab. The only normal tissue that has previously had its driver landscape analysed at epidemiological scale is blood, due to the propensity for driver mutations in hematopoietic stem cells to expand exponentially, allowing them to reach variant allele fractions that exceed the error rates of standard sequencing, as I've mentioned earlier. Reassuringly, when we looked at the blood target analysis data, all 14 driver genes we identified were already known to be associated with clonal hematopoiesis. However, this approach does show that standard sequencing approaches are only seeing the tip of the iceberg when it comes to driver landscape. Targeted analysis is able to detect drive mutations at much lower variant allele fractions, meaning we observed around 12 drive mutations per donor compared to the less than 0.1 mutations seen by standard approaches. Turning to the buccal swabs, the drive landscape of normal oral epithelium has not previously been described. We expected it to show similarities to skin and esophagus, as it is also a squamous epithelium, but it was unlikely to be identical, as the drive landscape of head and neck squamous cell carcinoma and esophageal squamous cell carcinoma differ from each other, and oral epithelium is derived from a different embryonic germ layer from esophagus. We discovered an unprecedentedly rich driver landscape with over 62,000 drive mutations and 46 genes identified as being under significant positive selection. Of note, several genes appear to be selected later in head and neck squamous cell carcinogenesis, including B2M, B2 microglobulin, and HLA-A, which may play a role in immune evasion, and also SMAD4 and CDKN2A, which interestingly also are late driver mutations in pancreatic cancer evolution. To exemplify how rich this collection of drive mutations is, I'm going to briefly compare it to two commonly used cancer sequencing databases. The Catalogue of Somatic Mutations in Cancer, which recently celebrated its 20th anniversary, is an invaluable resource that has attempted to curate all data generated from cancer sequencing studies to date. This plot shows that for all the most common driver genes, apart from p53 and oral mucosa, we have detected more mutations than the combined whole exome and whole genome sequencing carried out across all cancer types in COSMIC, so effectively the entire history of cancer genomics. This difference is even more pronounced if we solely consider squamous cell carcinomas from across tissues. The reason for this difference is that most cancer sequence efforts only detect essentially a single clonal expansion, whereas we are detecting hundreds of clones per donor. If the buccal swab samples were sufficiently polyclonal that we were very unlikely to detect the same clone twice, then the number of clones we've sampled would be approximately equivalent to half the cumulative duplicate coverage we've retained, which would be around 350,000 clones. So 2 orders of magnitude more than all of the squamous cell carcinomas have undergone whole exome or genome sequencing to date. To further emphasize this point, the Cancer Genome Atlas was a pioneering project that underpins much of our current knowledge of cancer genomics. It generated 7,500 cancer exomes from across 29 cancer types and identified a total of 15,000 drive mutations, which is a quarter of the number of drive mutations identified in this study. One piece of information I've slightly glossed over so far is how we actually identify driver genes. We normally do this by looking at the ratio of non-synonymous to synonymous mutations within a gene, after taking into account trinucleotide composition and genomic covariates. For a passenger gene that neutrally acquires mutations, we expect around 3 times as many missense mutations as synonymous mutations, and higher impact mutations such as indels and nonsense essential splice site mutations to account for on the order of 10% of all mutations. By contrast, for a gene under positive selection, we instead expect to see an enrichment of non-synonymous mutations compared to synonymous mutations. Activation of oncogenes primarily occurs via missense mutations at specific residues, whereas tumor-suppressing genes can be inactivated by a variety of missense mutations throughout the gene, or by nonsense mutations, essential splice sites, mutations, and frame-shifting indels. We can also in theory detect negative selection, where genes have a depletion in the expected number of missense or nonsense mutations. Negative selection is the dominant evolutionary pressure for germline mutations, but seems to affect somatic mutations relatively infrequently, only really being seen in essential genes in haploid regions of tumors. Consistent with previous results from cancer, we observed that the vast majority of genes exome-wide accumulate mutations neutrally. The positively selected driver genes have an enrichment of missense and nonsense mutations, but there is also some signal for negative selection within essential genes. Detecting negative selection at the individual gene level requires a very large number of mutations per gene in order to detect that there is a significant depletion of non-synonymous mutations. However, we were able to do so in a study. Of note, promoter mutations in TERT and hotspot mutations in SF3B1 and PIK3CA were under positive selection, but nonsense mutations affecting these genes were under negative selection. Turning to the mutation distribution within each gene, this plot shows the frequency of coding or essential splice site mutations observed at each amino acid in p53. The shading denotes the cumulative duplex coverage obtained across the gene. As expected for p53, missense mutations were primarily observed in the DNA binding domain, whereas essential splice site and nonsense mutations occurred throughout the gene. We can compare this mutation distribution to that observed across squamous cell carcinoma in COSMIC. In general, the mutation distributions are remarkably similar. with very good overlap between the observed mutation frequencies of canonical missense hotspots. There are, however, a few subtle differences. For example, we see a greater proportion of nonsense mutations in cancer than in normal tissues, which may reflect the ease of requiring a single dominant negative missense mutation rather than having biallelic loss during the shorter evolutionary of a normal tissue clone. As I'm sure this audience would expect, we observe dramatically different mutation distributions across some of the most frequently mutated driver genes. Notch1 is the most common driver and exhibits a very large excess of missense mutations in EGF repeats 8 through 12, which is the ligand binding region. We also see nonsense mutations distributed throughout the gene, although there is a notable lack of them in the final exon. We expect nonsense mutations in the final exon to cause C-terminal truncation rather than nonsense-mediated decay. So this pattern is consistent with the loss of function pattern observed in p53. By contrast, PPM1D, which is a phosphatase that removes activating phosphate groups from p53 and so keeps p53 inactivated, has a striking enrichment of nonsense mutations and indels within the final exon, which are expected to cause C-terminal truncation. This results in loss of a negative regulatory region of PPM1D and so results in protein stabilization and thus high levels of p53 repression. RAC1, a small GTPase, shows a pattern consistent with oncogenic activation. In skin cancers, we mostly see activation via proline 29 leucine mutations, whereas ARC data revealed selection of many other missense mutations spread throughout the primary sequence of the protein. However, reassuringly, when we map these onto the 3D structure of the protein, they all cluster around the GTP-GTP binding site, as we may expect, given that these mutations may leave RAC1 in its constitutively active GTP-bound form. We have developed two approaches for determining whether specific residues in a protein are under significant positive selection, using site or codon DNDS as one thing, and then the second I'll talk about later. We can identify significant recurrence of mutations at individual residues. Consistent with the previous plots, we see that all the significant single nucleotide variants of PPM1D cause nonsense mutations, whereas all of them in RAC1 cause missense mutations. Of note, we also observed a small number of synonymous hotspots, one in p53 at threonine 125 and 5 in Notch1. The p53 synonymous mutation hotspot has previously been observed by Ben Lenner and has shown to impact splicing. All 5 of the Notch1 synonymous mutations were predicted with high confidence by Splice AI to also disrupt splicing. We also developed a within-gene DNDS approach, which uses the large number of mutations within a single gene to build a background model. This found relatively little evidence of selection from many non-coding mutations, although there were a small number of intrinsic mutations under selection that primarily occurred to disrupt splicing, as well as a significant enrichment of mutations in the polyadenylation signal and transcription start site of p53. It's important to note that the enrichment mutations in the core promoter region for many non-driver genes mostly reflects an increased mutagenesis in the region rather than selection. This is consistent with previous work showing this phenomenon by Neurolopez, Bigas, and others. We do see genuine signals of selection in the well-known promoter mutations in TERT. In addition to the 2 canonical mutations that are commonly described with TERT, we also identified 3 other significantly recurrent promoter mutations that have previously been observed in cancer but are less well appreciated to be drivers. Perhaps of greatest interest to the audience, we can use this approach of detecting selection at the site codon level to inform whether variants of unknown significance in ClinVar are likely to have a functional impact on the affected protein. This is an area that we have only recently started exploring, so I hope this data set that we have generated is of use to the people in this audience with greater expertise in this topic. As shown in these plots, sites which are identified as being pathogenic, likely pathogenic in ClinVar, have significantly higher DNDS ratios, with many of them reaching significance after FDR correction. Additionally, we identified many variants of unknown significance that have comparably strong evidence of slight level selection, indicating that they are very likely to alter protein function in a similar way to known pathogenic variants. Reassuringly, no mutations identified in ClinVar as being benign were close to reaching significance by site DNDS, supporting this approach does correctly stratify mutations by functional impact. Large-scale genome editing approaches achieved by CRISPR and prime editing have recently made in vitro saturation mutagenesis studies possible. In many cases, these use HAP1 cells to avoid the complications of a second allele, and they mostly use cell survival as the readout. One nice feature of our data is that it can tease out much more subtle signals of selection that may have gradually accrued over decades. SBS5, which is a mutational process common across all cell types and accounts for a substantial amount of the intrinsic accumulation of mutations with age, is able to generate single nucleotide variants at all trinucleotide contexts, which means all S and V types can be profiled simultaneously. Furthermore, the frequency of the mutation classes corresponds to the mutational probability in vivo, which means we are more likely to reach saturation for biologically relevant mutation types. However, there are also several disadvantages of this approach compared to more traditional in vitro saturation mutagenesis approach. We can't choose a particular gene to study. We're limited to only being able to study genes which have relatively strong positive selection in a given tissue. Also, tissue choice may identify only a subset of driver mutations. So, for example, Notch1, as I've shown in all mucosa, is frequently lost. It has common loss of function mutations, but in blood, you often have gain of function mutations in Notch1, which manifest as missense mutations in a different region of the protein, or C-terminal truncation losing its PES domain. We have also not quite reached saturation in this study. We need double the data set to have on the average of 1 mutation per amino acid. We're unable to assess all mutation types. We can't see structural variants, copy number changes, or large indels by targeting NanoSeq. And we also can't tell which clones have multiple drivers. And then finally, we only provide information about when the phenotype is affecting cell fitness. We can't see other phenotypic consequences. Moving on from variant impacts, our data also provides other insights into clonal dynamics. As previously mentioned, we know from clonal hematopoiesis studies that in blood, clones can grow exponentially until they may ultimately cause a complete selective sweep of the bone marrow, as shown in a nice preprint by Jamie Lundell. Our data is entirely consistent with this observation, with the aggregation number of blood cells containing DNMT3 mutations increasing exponentially with age, and most of this is driven by a single large dominant clone. This is in stark contrast to the behavior in oral epithelium. We generally see a linear increase in the aggregation number of buccal cells containing a drive mutation, but the size of the largest clone remains very small. This suggests there is some mechanism that constrains clone size. Of note, this is not likely to be interclone competition, because we estimate that only 10 to 20% of cells in oral epithelium contain a driver by late middle age. For those unfamiliar with this style of plots, in order to extract mutational signatures, we often classify single nucleotide variants into 96 categories, determined by the 6 mutational classes, annotated by the pyrimidine base, so cytosine to adenine, cytosine to guanine, cytosine to thymine, thymine to adenine, thymine to cytosine, thymine to guanine. These are then each split into 16 categories, corresponding to the trinucleotide context surrounding the mutated base. We identified 2 common mutational signatures in oral epithelium. These are both similar to previously described mutation processes seen in other normal tissues in cancer. Signature A, which is a mixture of age-associated signatures to SPS1 and SPS5, and signature B, which corresponds to SPS16 and is associated with alcohol consumption. Of note, we did not observe any of the common signatures associated with smoking that have been seen in lung cancers, SPS4 and SPS92, or any evidence of apoplectic mutagenesis, SPS2 and SPS13. The absence of the smoking signatures may be due to low expression of CYP1A1 in oral mucosa, which is the main metabolizer of benzoate pyrene, or the differences in the structure of oral versus respiratory epithelium that may protect the basal stem cells from exposure to benzoate pyrene. As expected from what I've just told you about the already known associations for these mutational processes, we observe that signature A accumulates linearly with age, whereas signature B causes high levels of mutations within heavy drinkers. One thing that surprised us about signature B is that one thing that surprised us about signature B was its propensity to generate driver mutations. We see that it is mostly enriched in non-coding regions, and it also accounts for a modest number of driver mutations. As shown in the lower plot, this may be partly due to its inability of this mutational process to give rise to nonsense mutations. Another advantage of our targeted NanoSeq approach is that it enables us for the first time to carry out studies of somatic mutations at epidemiological scale. We used multivariate mixed effect regression models using major risk factors and other relevant metadata as covariates, and different measures of mutation rate signatures or driver density as outcome variables. As may be expected, age strongly associates with S&V burden, specifically via signature A, and increased driver density for all of our driver genes. Somewhat surprisingly, we did not see any particularly strong association with sex, which may suggest that the high levels of head and neck squamous cell carcinoma may primarily be due to differences in lifestyle factors. As mentioned on the previous slide, drink years were strongly associated with signature B and had relatively low potential to generate drivers, and whereas smoking showed an association with both signature A and B, suggesting it is capable of accelerating age-associated signatures and exacerbating the effects of alcohol. Interestingly, we also see strong association between pack years and Notch1 driver density, and this suggests it is capable of altering the selection of mutant clones in addition to its mutagenic effect. Very briefly, given that we had a twin cohort, we looked to see whether there was any heritability of these phenomena of either mutational process or drivers. For signature A, which is age-associated, we do actually see that monozygotic twins, MZ identical twins, show more similarity from residual difference in burden analysis than either dizygotic, non-identical twins or unrelated age-matched, sex-matched individuals. Whereas signature B shows no heritability, which is what you may expect for something that's primarily driven by lifestyle factors. Intriguingly, both the driver fraction for Notch1 and P53 do show quite clear evidence of heritability, which is consistent with that work that I showed previously in the bladder, of there being individual driver preference. We still need to get to the bottom of this phenomenon. Zooming out, I hope I've convinced you today that our approach has some use for exploring the molecular basis by which cancer risk factors actually increase cancer risk rather than it being a black box that it has been to date. We argue that most of these risk factors will likely have an impact on the clonal landscape observable in normal tissues, and that we can use this in the future to make quantitative predictive models for cancer risk. This may allow us to understand the molecular role of known risk factors, such as obesity. It also means that we may be able to answer questions about the likely impact of novel exposures, such as vaping, without necessarily having to wait decades to see the impact on cancer rates in traditional epidemiological studies. It also gives us the long-term hope that we're able to modulate these clonal landscapes pharmacologically to reduce the risk of cancer. Here are a few take-home messages from my talk. I hope you've been introduced to what is a hidden world of somatic evolution within our tissues, that mutations accumulate in all somatic tissues, largely independent of whether there is cell division, to have on the order of 20 to 50 mutations per cell per year. Clones of 1 to 2 drive mutations are common in many dividing tissues, but the genes and their frequency varies dramatically, greater than 50% of esophagus and endometrium, 10 to 20% of bladder and lung, less than 0.5% in pancreas. These results are entirely consistent with a multi-stage somatic evolution model of carcinogenesis. These clones are multiple steps away from being cancer cells. We would expect to see some clones with the first driver. And also carcinogens and risk factors can induce both mutations, mutagens, or selections. Finally, this is my most important slide. This work was only possible due to the work of many people, including those in the Martin Craner Lab, cover groups in Sanger, and as well as our collaborators in Twins UK and Cambridge Biopositive Translational Medicine. I'd also like to thank our funders at Wellcome, Cancer Research UK, and Joseph Sainer, and also thank you for your attention. I'm happy to answer any questions, or we may not have time, but it's...
Thank you. We do have time for questions.
Hi, thank you for the wonderful talk. I just wonder, how did you exactly perform the deconvolution of the signatures, and how good or not that good was the Kazan similarity for your approach?
Yes, so as you see in the final paper, we leave them as signature A, signature B, rather than doing a full deconvolution. We did do deconvolution using both sig profile and HTP, and we did various different rounds of applying either priors for existing signatures to see if there's any novel signatures. We're fairly confident in the result that there aren't low levels of SPS4 smoking signatures or SPS2 and 13. We thought it was more simple and straightforward to show it as the extracted single signature rather than forcing it to be split into SPS5 and 1, but it does end up with a greater than 90.9 cosine similarity if we do split it into.
Sorry, well, do you use the web version of SIG Profiler or Python or R? It actually really matters.
Oh, okay. we can discuss that later, because yes,
you have shown in one of the many slides that you have some differential expression in male and female that seems to be, at least in the list of driver genes, and it seems that at least in some of the cases the difference were pretty impressive. So are those small numbers or are those real?
So
comment?
I think you're referring to the bladder work where there was the individual preference. So one of the things from that study was, unfortunately, we only had 15 donors, right? And so it's worth noting that the bias that you picked up on very astutely, that in KDM6A we seem to have a greater degree of enrichment in women than men, is consistent with what is seen in non-muscle invasive bladder cancer. So it likely is real, but this has been followed up by work by Nuro Lopez, and it looks like there are some quite interesting, significant differences based on sex.
Thank you. What's your opinion on the relevance of somatic mutations in non-cancerous diseases, actually? Because you focus on driver mutations, do you think that those are the main that have any relevance for phenotypes and eventually will end up in aging or faster aging or cancer? Or do you think there could be like stable driver mutations that clonally expand and still have some relevance to non-cancerous phenotypes?
So, I mean, that is very prescient. That's looking forward to what the next work that our group is mostly doing, of trying to understand what diseases may be affected by somatic mutations. We have found some quite exciting results, which we can't quite share quite yet. Having said that, it was pretty remarkable to us that the esophagus phenotypically behaves so similar to how it should in mouse models and in culture when it is very highly mutant. So in some cases, I think a relatively small number of somatic mutations or small number of drive mutations can have a big tissue level phenotypic effect, but some tissues, the benefit may be happening at the level of the stem cell just in terms of differentiation without having a major effect on how the tissue phenotype behaves. So there's still a fair bit of work to be done.
All right, I think for the sake of time, we're going to move to the next speaker. Thanks again.